Hi, so we're looking at cellular respiration, the entire system that you need to know for that in the Metabolism and Survival unit. Okay, so respiration you do know a reasonable amount about from Nat 5. You should know it's a series of enzyme-controlled reactions that release energy from food. That's still the case. Okay, every single living thing will have respiration pathways because you need to release energy from food. And without them, every energy that is in food couldn't be converted in a form that we can use. And to be very clear, as it is, food is not useful to us. We need to get that chemical energy, that potential energy, into a form that we can use. That's ATP. A huge part of this now is understanding how that happens. You should already know that ATP stands for adenosine triphosphate. You should know that it's used to transfer energy back and forward between the respiration pathway that generates it, which actually now you should recognise would be any catabolic reaction, to any reaction which requires it, which again are your anabolic reactions. ATP, when it's drawn for you, is unlikely to be shown in its full chemical structure, but it could be. You would normally have it shown as an A with just 3 phosphates attached, and quite often that extra bond in the second and third is going to be shown slightly differently because that is the high energy bond, but they don't have to show it that way. So inside our chemical reactions there are lots of things that we move commonly, and so we need to have terms that we're using for them a lot. We move phosphates an awful lot, so obviously linking to ATP will be a phosphate movement. That'll be from ADP and BiH to make it ATP, so that is phosphorylation of ADP to get ATP. So phosphorylation is going to be mentioned quite a bit. We shift hydrogens quite a lot, and the enzymes that remove hydrogens are called dehydrogenase enzymes. You're going to need to know that. They also move high energy electrons off at the same time. More detail in just a little bit. And then we also move carbons an awful lot, which is not surprising. That's generally our food sources. These are decarboxylase enzymes that will remove carbons. Our main substrate for respiration, and the only one that you're going to need to know in detail, is glucose. Okay, it's a simple monosaccharide sugar made during photosynthesis. It's nice for us in terms of how we can break it down. We get a clean breaking it down into water and carbon dioxide. You do not need to know full structure. You are, however, expected to know that it's a C6 in terms of its size. I would expect that you know it's a carbohydrate, so water is H2O, so that tells you in terms of ratio, that's why it's a hydrated carbon. Right, so to our first stage. So first stage is common for everything. You already know this. This is glycolysis. Okay, it's in the cytoplasm. We're going to take a glucose molecule, which I've said you need to know the carbon number 6, and we're going to break it down into 2 pyruvates. Since we've broken a 6 into 2, obviously they must be 3s. It's what you already knew. So what we're doing is taking glucose and breaking it down into pyruvate. You will notice with this diagram it's 10 steps. So when you were doing this in Nat 5, you just went glucose, line, pyruvate. Now you need to go glucose, several stages, pyruvate. You don't need to know it's 10 stages, you just need to know there's several. Right, so let's bring glycolysis up to higher. So here's our glucose to pyruvate. But that's not true. We need to put it as a series of intermediate stages running down to the pyruvate. So we've got several different stages. You don't need to know the names of any of the intermediates. You just need to know they're called intermediates. Now, at this point, we need to bring in some energy, and we're going to phosphorylate some intermediates using ATP. We need to use 2 ATPs to do this, so this is an energy investment stage. And the reason it's called investment is because we actually then have an energy payoff where we get enough energy out to generate 4 ATPs. So although we paid in 2, we got out 4, so we have a net gain of 2 ATPs from this stage. Now you knew there was 2 ATPs from Nat 5. You now need to know this: energy investment, energy payoff, and the term phosphorylation of the intermediates. Not quite finished, because at the same time as doing this, what we also managed to do is to pull off some hydrogen and some high energy electrons, and they're picked up by this particular chemical, NAD, which is called a hydrogen acceptor. So it picks up those hydrogen and the high energy electrons to make NADH, and that's all going to be done with a dehydrogenase enzyme. Okay, this is now your glycolysis. Okay, so we've now got pyruvate, and pyruvate is super nasty. This is not a chemical that we can have lying about in our cells. So if you don't have any oxygen, you're going to pass it along into fermentation pathways. You're going to get a much lower yield of ATP. Basically, you're just going to get that 2 that you managed to get from the top. So pyruvate in animals will go down to lactate. The nice thing about that for us is that it's reversible, but we're going to get an oxygen debt for every one that we do, and then we can take it back up if oxygen appears. In plants and yeast, ethanol and carbon dioxide. The ethanol is held, but the carbon dioxide is released because it's a gas, so it just diffuses out, so that is irreversible. Nothing more than you had at NAT5 here. Now at NAT5 we just said, okay, your pyruvate goes into the mitochondria, set of reactions, get out a whole pile of energy and some water and some carbon dioxide. That is definitely not enough detail for the higher. So what we have first of all is a transition step. So we're going to say there is oxygen, so I'm ready to move into mitochondria, but pyruvate is not the one that we can use to feed into the cycle, and the cycle we're trying to get into is the citric acid cycle. So inside our mitochondria, we're going to take the useful bit of pyruvate. So what we need to do is take pyruvate and break it into a 2 rather than a 3. So we're going to lose 1 carbon, that's carbon dioxide. At the same time as we did that, we're going to get some hydrogen ions and some high energy electrons. So NAD is going to come in and pick this up. The reason we need to use these carriers is because hydrogen is way too exciting to have as free hydrogen, like it's explosive. So you need it to be picked up by something that can kind of nanomolecule it over to where it needs to be. Okay, so your acetyl is now the right size. We need a 2 carbon to join on to a 4 to make this citric acid cycle work. But it's not good enough the way it is in terms of moving. So what we need is for it to be picked up by another carrier. The carrier for this one is coenzyme A. And when acetyl and coenzyme A join together, you get acetyl-CoA, or acetyl coenzyme A. Now the acetyl coenzyme A is going to take it into the cycle and attach it to a compound called oxaloacetate. And then once that's done, it's going to go back and pick up another acetyl. So the coenzyme A is another carrier in the system. Okay, so here's your full citric acid. Central matrix of the mitochondria, so that's in the kind of liquidy stuff inside it. Our 2 carbon acetyl joins onto the 4 carbon oxaloacetate, and that makes a 6 carbon because 2 plus 4 gives us 6, and that would be citrate. And then the citrate gets broken down again back to oxaloacetate. So we take a 6, drop off a carbon gets us back to a 4. That's the basics. As we're going to get more of this dehydrogenase enzyme action. So we're going to remove hydrogens and electrons. They're all going to be picked up by that NAD to make NADH, and we'll get rid of those carbon dioxides. We'll get an ATP, but just 1. Okay, to show this in diagram form. So here's our acetyl-CoA, here's our oxaloacetate. I'm afraid you need to know all these terms. So this is a 4 carbon, 2 carbon is going to join onto it, the CoA is going to go away and pick up some more acetyl. The 2 plus the 4 gives us 6. This is citrate. We're going to get rid of 2 carbon dioxides. We're going to make an ATP. We are going to also pick up some more hydrogens and high energy electrons by NAD. And the whole point is to get that NADH. Okay, at this point here, you've still not got a lot of energy generated. You've got 2 from the top and 1 from in here, which, okay, you had 2 acetyls because you had 2 pyruvates. That is not a lot from a glucose. So what you really need is the last stage. Okay, your final stage, electron transport chain. This is where you actually get the energy out of the rest of it that you were trying to do in the first place. So you're going to use the inner membrane of the mitochondria this time. This is called the cristae. And this cristae is the folds. And the folds are there to increase surface area, because the greater surface area you have, the faster your reaction can be. You've got 2 really important sets of proteins on here. You have hydrogen pumps and you have ATP synthase. Now from the name, ATP synthase must be synthesizing ATP, making ATP, and hydrogen pumps must obviously be pumping hydrogen. Now if they're pumps, that means they need energy, and we're going to use those high energy electrons that have been taken out with the NADH and use that to drive the pumps. Okay, so the high energy electrons move along the chain, and as they go, the pumps are effectively charged up to move the hydrogen ions, which have also been dropped off by the NAD. Okay, so what happens is inside the link between the two membranes, so the gap in there, this will become super packed with hydrogen ions because of these pumps moving it in. That's not the best colour for it, sorry. The pumps moving it in across it, driven by those high energy electrons. So the hydrogen has been delivered and then it's been pumped into the gap, creating, and this is really important, a high concentration of hydrogen inside the gap. So to go through this again, your electrons are being used to power these pumps, these hydrogen pumps. They're going to move the hydrogen into the gap between the inner and the outer membrane. What you've now got is a high hydrogen concentration on the inside of this space, and on the outside of it, which would be inside the mitochondria in the central matrix, it's going to be low. That's going to be able to drive something, that's going to drive the ATP synthase, and then we're going to end up with hydrogens, and our electrons that have finished moving along the chain, they're going to be picked up by oxygen and they're going to make water. Now you need to know that when you're making water, it's the hydrogen ions and the high energy electrons, not just the hydrogen to make the water. Another little diagram to show it. Don't worry about this other carrier here, FAD. You will see it in some textbooks. The only one you're required to know is NAD. So the whole point of this was to make ATP. And you're going to use that enzyme complex that I've already mentioned on the Christie called the ATP synthase. And how ATP synthase works is driven by this hydrogen ion concentration. So this would be in the gap between the inner and the outer, and this would be in the central matrix. Our high hydrogen ion concentration here, and it's low here, so it's going to push through, driving this, and what this does is basically turns this gets twisted round and round, and as it twists it manages to connect the ADP and PI into ATP, and that's you finally used up the energy that you started with in your food. Okay, you need to know all of that and all of the detail. You might need to go through it a few times.